Good morning. Wonders of the Animal World, lecture number 3. Subject Zoology, FY BSc semester first, paper first. Mechanism of formation of pearl. Few specific bivalves have this unique capacity of formation of pearl. Pearl is in fact deposition of the layer of nacre around a suitable nucleus. The nucleus is a small inorganic bed-like particle or even a parasite enclosed in living tissue. The deposits around the nucleus are made up of calcium carbonate and some organic compound like protein, fat, or carbohydrates. Pearl formation in mollusca is a response to injury. The foreign particle or the parasite causes damage to the mantle, and to repair this damage, the nacre is deposited over this particle which finally forms pearl. More and more layers of the nacre can be deposited over the nucleus, leading to increase in size of pearl. In natural pearl formation, the mantle epithelium extends out to form a pearl sac. It develops further just the same way as formation of a cell and ultimately result into the formation of a pearl. See the diagram. Mother of pearl, nacre, foreign body, like a sand particle or a parasite, mantle epithelium and mantle. Then layer of nacre is formed, and finally the natural pearl is formed. Bioluminescence definition. Bioluminescence is defined as the production or emission of light by living organism by using a chemical is known as bioluminescence, or bioluminescence is defined as emission of light by living organism through a biochemical process is known as bioluminescence, and the animal which is produce light are known as the bioluminescent animals. Sense of sight is generally not useful in the dark. Some animals generate light by the luminescence through the biochemical mechanism. This requires comparatively less energy and no heat is generated, hence, it is known as a cold light. Bioluminescence is used for the various reasons like the attracting mate, locating prey, driving away the predator, finding the path, etc. Some of the example of animal which is shows the bioluminescence mechanism. First, Noctiluca. Bioluminescence is the widespread phenomenon in nature. It is particularly predominant in the marine environment. Marine bioluminescence is mainly blue colored. In this case, light is produced by the chemical reaction in the presence of oxygen involving a substrate termed as the luciferin and the enzyme known as luciferase. The bioluminescent species of the Noctiluca is Noctiluca scintillans. In this unicellular organism and the organelles called as the scintilon is present which contain luciferin, luciferase and sometimes luciferin binding proteins. These thin vesicles are more dominant near the cell periphery. The scintilon are the activated in the condition of the stress. The various functions of bioluminescence performing in Noctiluca are as follows: recognition of the organism of its own species, oxygen defense, defense from predators. Second example, glowworm. Glowworm, Coleopteran insect, inhabit moist or the damp caves and cannot survive under dry or very low humid conditions. This larva stage molt 4 times before entering the pupal stage. This may take a few molts. Pupal stage lasts just above a week and then metamorphoses into the adult fly. The adult fly also is short-lived. Males stay near the female pupa for it to hatch and then mates. Female adult after mating lays over 100 eggs at a time which hatch within 7 to 9 days. If the evolutionary development is taken into the consideration, the ancestor of these were herbivores feeding on fungi. Gradually, they modified the snares into insect trap and become a insectivorous. Third example, firefly. Fireflies shows the bioluminescence for the attracting the mating partner, generally only males. The flashing in males follow a particular pattern which is specific for the species. Female fly can identify the species on the basis of the pattern of the luminescence to select the mating pattern. The luciferin in fireflies is called as the Photinus luciferin. ATP undergoes the pyrophosphate cleavage and form a luciferyl adenylate. Molecular oxygen reacts with it in the presence of the enzyme luciferase. This is oxidative decarboxylation of the luciferin and results into the oxyluciferin. Light energy is emitted during this reaction. Color of this light depends upon the molecular structures of the luciferin molecule. Example 4, anglerfish. In anglerfish, sometimes the phenomenon of the bioluminescence is used for the predation. The most famous predator to use the bioluminescence could be the anglerfish that use the bioluminescence to lure prey. The anglerfish has a huge head, very sharp teeth, and a long, thin, fleshy growth known as filament on the top of its head. At the tip of filament is a wall called as a isca that the anglerfish can light up. Smaller fish, curious about the spot of the light, swim in for a closer look. The light is due to the bacterial bioluminescence at a isca. Thank you.